how do you go about proving viral causation? 100% of the people who just a minute ago told me that they're sure this is caused by a virus don't even know how you would go about doing that. In order to understand this, what's called COVID-19, we need to be very clear on terms and what we're talking about. COVID-19 is the name of the disease, which is supposedly a new disease. And the coronavirus, otherwise known as SARS-CoV-2, is supposedly the virus that causes COVID-19. So in order to understand this, we have to understand how a biologist, a virologist, or a medical doctor proves causation with a virus. That's the first step. So we should go back and say, how did the medical community determine that the disease COVID-19 is caused by this new coronavirus? So the first step is, when I ask people that, and I'll say, why do you think that this coronavirus is causing COVID-19? They usually say things like, well, there's a lot of people who died in New York City, or a lot of people who died of China, and then it spread to North Korea. Or there's a lot of people in a nursing home, or my Aunt Bessie went to a party and then she got it. And then on and on and on. So the problem is, these are what are called epidemiological observations. And there is not a reputable scientist or medical doctor in the world who thinks you prove viral causation with epidemiology. In other words, if you or anyone out there thinks that the proof that it's a disease is caused by a virus because a lot of people in the same place died of a similar disease, then you must think that Hiroshima was a virus. Because I can tell you a lot of people died in the same place of the same thing, and as far as I know, nobody thinks that was a virus. So then you could say, well, yes, but what if it spreads? That proves it's a virus. So there was also an event called Chernobyl, where they blew off a nuclear reactor, and then it started spreading all over Eastern and then Western Europe, and thousands and maybe millions of people got sick with the same thing. And so this spread, and as far as anybody thinks, nobody thinks that's a virus. Here's another example. A lot of sailors on ships, one after another, they had their teeth fall out and then they died of heart failure. And for 200 years, they thought that must be caused by some spreadable microbe that they couldn't see. And then one day somebody ate a lime and the whole thing went away because it was scurvy. And the point is, you cannot prove viral causation with epidemiology. And it has consequences if you're wrong. We thought beriberi and pellagra, which killed millions, was caused by an unseen microbe because the epidemiology was there. A bunch of families got it, and then the next family got it. And it turns out it was a nutritional deficiency. So there is nobody who thinks that epidemiology proves viral causation. Nobody. And anybody who says, I think this is a virus because Uncle Fred went to a party and he got sick, if they say that, they're basically scientifically illiterate. And I have gotten to the point where I say we have enough epidemiological evidence with COVID-19 that we should investigate a microbial cause. Now, we also have enough epidemiological evidence to investigate many other causes like electromagnetic field toxicity and air pollution and a bunch of other things. None of those prove that those are the cause either. But the purpose of epidemiology, of these observations, is to generate hypotheses that you can test in the lab. Okay, so now that we've got that clear, so I don't need anybody to tell me more epidemiological observations. We've already decided that we're going to investigate viral causation. Now, how do you investigate viral causation? It's interesting that all these tens and maybe even hundreds of people who tell me they're sure that they know that this is caused by a virus, when I ask them the simple question of how do you go about proving viral causation? I mean, I could ask you that. Do you know? No. I've not met one person who even knows actually how to answer that question, let alone knows what the answer is, which means they have no idea. Now, let me just say that again. 100% of the people who just a minute ago told me that they're sure this is caused by a virus don't even know how you would go about doing that. Now, that should be shocking to people, because if you're listening and you don't know how to do it, then you have no idea whether it's been done or not. By the way, I didn't know this as well as I do now until about a year ago. In fact, I've said things and written things which were incorrect because I didn't know the science of this as well as I should have. And the only reason I point that out is I can pretty much guarantee that your family doctor and your internist and your surgeon doesn't know this either. Now that may be shocking to people, but I didn't learn any of this in medical school or any training. This one has to figure out for themselves. So here's the way. First of all, you have to isolate the virus. So take an example of, we want to do an experiment, and please listen carefully to my words here. We want to do an experiment to see if the caffeine in coffee beans causes high blood pressure, right? So let's do an experiment, and I'm not going to say this each time, but let's say we do proper controls, proper placebos, everything about the study is good. So we take coffee beans and we grind them up and we put them in a capsule and we give them to 10 people, and all of their blood pressure goes up and none of the placebo do. Have we now proven that the caffeine in coffee beans causes high blood pressure? The answer for any rational person would be no. Because unless you think the only thing in a coffee bean is caffeine, which you can prove isn't correct, there's fiber and caffeic acid and aromatic oils and probably 100 other things, you have not identified that it's the caffeine that causes high blood pressure. Okay, so let's take it a step further. Let's, like we do with coffee, we put it through a filter paper and we put water through it and we drip it through, and that catches the ground part, right? And you throw away the ground part, and now you know, because you know that caffeine is water soluble, you know that the caffeine is in the liquid part. So you give 10 people the coffee, 10 people controls. Have you now proven that it's the caffeine in the coffee beans that causes high blood pressure? The answer of any sane, rational person would be no. Because again, there's a lot more things in coffee than just caffeine, obviously. There's flavor and oils and all minerals, etc. Okay, so we want to be very clear and precise because we're talking science here. So we do some technique, and there are many of them in modern chemistry and modern biology, where you pull out just the caffeine from the coffee, right? So you can do it through a centrifuge and it comes out in a certain band. You suck the band out. The next thing is you have to prove to people that the only thing you have is caffeine, right? So you do a chemical analysis. There's no caffeic acid. There's no aromatic oils. All you have is pure chemical caffeine. And you publish, this is the steps I did. This is all we have. And you publish the independent analysis. This lab has said in this sample, all there is caffeine. You give it to 10 people, their blood pressure goes up. Have you now proven it? The answer is yes. That's how we do science. Now, the reason I say that is because that's almost exactly how we isolate and purify a virus. And where am I getting this from? Well, I'm getting this from 20 years of research from about 1935 to 1954, where they did this exact procedure. They took people like with chicken pox, and they ground up their snot or their vesicle, fluid, and they had the basically macerated cells. Now that's not the virus any more than that's caffeine. So then they put it through a filter, and the filter would catch the bacteria and the cells and tissues and all the big stuff, and then you would have the virus and a lot of other things. That's still not the virus. Then they would put it in a centrifuge, which separates things by weight, and the virus, because it has a certain weight, separates out into a band. And then they would suck out the band, and they would show you with an electron microscope picture, this is all we have, pure virus. Then they would expose an animal to that virus, and none of them got sick. 20 years. They exposed every type of, quote, viral illness they could find. They isolated and purified the virus. No animal or human got sick, and they said basically there's nothing to this. Now, here's the shocking thing, and I'm about to sort of prove it to you. Nobody has isolated a coronavirus from anybody with COVID-19. How do I know that? Let's say what the CDC says about this. These are excerpts from published papers. The first one is from July 13th, 2020. It's from the CDC 2019 Novel Coronavirus Real-Time RT-PCR Diagnostic Panel on 39. So this is their report on the state of science of what we know about this COVID-19 and coronavirus. They say, quote, since no quantified virus isolates of the 2019 nCoV virus are currently available. So what does that mean? It means the CDC, as of July, has never isolated the virus. I remind people that a virus is a thing, right? Like a fork. I usually have a fork, but I don't have a fork. We're not talking about a thought or a feeling. We're talking about a thing, like a chair or a fork. Every fork can be isolated. Every fork, if I have 3 forks, can be quantified. This is 3 forks. If you say no quantified viral isolates are available, you've never isolated the virus from anybody with COVID-19. Now, are they the only ones? So here's an excerpt of the paper called Detection of the 2019 Novel Coronavirus by Real-Time RT-PCR. This is the paper published in a peer-reviewed journal called European Surveillance that inaugurated the pandemic, so-called. This was by Christian Drosten and a bunch of other virologists, and they were tasked with developing a test for this new coronavirus. So let's see what they say. This is quoting from the paper. The ongoing outbreak of a recently emerged novel coronavirus poses a challenge for public health laboratories as virus isolates are unavailable. In other words, they were tasked with making a test for a virus, and they have no examples of the virus to start with. That's like if I asked you to make a test for a unicorn, you might ask me, can I see a unicorn? And I might say no. And you might say, well, how am I supposed to make a test to find a little piece of a unicorn when I don't know what a unicorn looks like or what a unicorn is made of? And then I might say to you, here's $1 million, do it anyways. And then they go on to say, quote, we aim to develop and deploy robust diagnostic methodology for use in public health laboratory settings without having virus material available. They had no virus to make a test to find a piece of the virus. Therefore, their PCR test The test, which is being used all over the world to document cases, is a complete fraud. They have never isolated a virus and said, this is a unique piece of that virus, because they have no virus. Now, are they the only ones? Here's a quote from Pfizer about making their RNA vaccine. To build an RNA vaccine, scientists do not need the actual virus. In other words, Pfizer admits they have no virus with which to make a vaccine against. They call it an actual virus, as opposed to, I guess, a theoretical virus. Now, I would change the word theoretical to say an imaginary virus, or a make-believe virus. So they're using a make-believe virus, which they have never isolated, they don't know the sequence of, they don't know the components of, to make a vaccine against this virus, and God only knows what they're making a vaccine against. Here's another one, a paper published in Nature, which was one of the first papers on describing the novel coronavirus. And a friend of mine named Torsten Engelbrecht asked him, because he says he isolated the virus. And people have sent me about 20 different peer-reviewed articles claiming they isolated the virus. When you look at the paper, it's very clear that nobody has ever isolated the virus. It's a fraud. And we asked him, did you isolate the virus? He said, quote, we did not obtain an electron micrograph showing the degree of purification. In other words, they didn't do what every peer-reviewed journal article has to do, which is say, here's the steps we did, here's the picture showing that we have an isolated virus. They didn't even take a picture. And they admit they didn't isolate the virus. Here's another one. Same thing. Identification of a coronavirus isolated from a patient in Korea with COVID-19. This was published by the Korean Center for Disease Control and Prevention. Essentially, it's the CDC of Korea. So we asked him, did you isolate the virus? Because that's what it says in the title. And the answer was, quote, we could not estimate the degree of purification because we did not purify and concentrate the virus. In other words, they have no virus. Here's another one. Again, isolation of the new virus. Quote, we show an image of sedimented virus particle, not purified one. The Canadian Health Ministry, in Freedom of Information request, do you have any information on an isolation of this virus? Their answer, quote, having completed a thorough search, we regret to inform you that we are unable to locate any records responsive to your request. None of these groups who are organizing this have any example of an isolated virus. If you've never isolated the virus, you have no way of knowing whether it causes disease. Period. And the test to identify a virus that you've never isolated is not false positive or false negative. It's just good old-fashioned false. This is a scientific fraud. And interestingly, just 3 days ago, a group of European virologists and pathologists dissected this Drosten study and said the basis of using this study, which is the basis of all the PCR tests, all the testing, is pure scientific fraud, and they demanded that this journal retract the article.